Hello from my side. I think after Daniel Drucker began his talk by saying that there's little window for this session to provide some new input, I hope that I can still do that for you. I'm very happy to be able to present some of my PhD work in front of you today on Anja's behalf, actually. I'd like also to thank the organizers for giving me the opportunity today. As already been pointed out, we've unraveled a new trafficking mechanism for the glucagon receptor by VPS37A that is then regulating glucose production. We in the lab of Anja Zeiger are trying to combine endosomal trafficking with metabolism. And these are two fields that are generally studied in separation, but we're trying to combine this. And to do that, we are especially interested in different components from the endosomal system, how they affect metabolism. And by doing this, we're trying to bridge scales basically from our knowledge that we have on molecules and organelles all the way then to tissue and organ physiology as well as animal physiology. As the previous slide already indicated, we're mainly interested in the liver, which, as you all know, is a very central organ for whole body homeostasis, which is regulating and for which it's already been shown that the endosomal trafficking system plays a substantial role in regulation of metabolism. The liver is, during fasting, involved in processes for energy production, and during refeeding provides functions for energy storage. But in order to switch between these two statuses, there needs to be a metabolic switch occurring that to a large extent is regulated by glucagon and insulin, as you all know. But this switch needs to be very fast. Upon the changes in nutritional status, within a few minutes, this switch needs to occur. So the immediate effects of that are most likely not due to transcription, but there need to be some quicker mechanisms in place. And one such mechanism can be provided by the endosomal trafficking system. As you might know, basically every transmembrane protein that is present at the plasma membrane is eventually being endocytosed, and that is to that extent that every 20 minutes the whole membrane proteome basically is refreshed just by internalization that is undergoing constantly. Upon internalization of, for example, receptors at the plasma membrane, they go through different endosomal compartments until at some point there's a decision point of whether this protein is sent back, so recycled back to the plasma membrane, or whether it's sent for degradation. And this fate decision basically provides a very quick and versatile mode for the cell to differentiate, to alter the proteins present at the plasma membrane upon, for example, nutritional cues. Whether or not a receptor is recycled or degraded is, for example, dependent on signaling motifs in the cytoplasmic tail of the receptor, or based on the ubiquitination status. And my talk today is focusing on this end of the image, where the endosomal sorting complex is required for transport, or ESCRTs for short, are mediating the engulfment of still active signaling receptors that are still signaling from endosomal membranes into early endosome, which is then leading finally to signal termination and the degradation of that receptor. And despite the interesting function of the ESCRTs in downregulation of receptor signaling, and its implication that this could have on metabolic regulation, there's so far very little research done on the ESCRTs in relation to metabolism. And in fact, there's only one publication so far that showed that activation of ESCRT complexes is preventive in the development of NASH by degradative pathways activated through this for the TLR4. And specifically, the authors in this paper suggested that the ESCRT1 complex might have a potential function in metabolism. So naturally, we wanted to understand this a bit further, and therefore we screened for mRNA levels of ESCRT1 subunits within livers of patients with either obesity or type 2 diabetes, and we found the complex subunit VPS37A to be downregulated in those livers, as well as a negative correlation of VPS37A mRNA level with both BMI and fasting plasma insulin. So based on this data, we then wanted to study this in mice and see what depletion of VPS37A is actually doing. And for that, we utilized lipid nanoparticles that are encapsulating as iRNAs, which, if we inject them into wild-type mice by tail vein injection, result in hepatocyte-specific depletion of the protein of interest. And doing so, we have then mice that are lacking VPS37A just in the liver. And when we phenotyped them for glucose tolerance or insulin tolerance, we did not observe any changes. However, when we treated those mice with pyruvate in a pyruvate tolerance test, we found significantly enhanced gluconeogenesis. And in line with this, we also found in livers of those mice enhanced gluconeogenic gene expression, as well as enhanced CAMP and decreased liver glycogen content. When we now injected those mice acutely with glucagon, we found enhanced phosphorylation of pKa substrate, both at basal state and at glucagon-induced state. And in addition to pKa phosphorylation, we also found enhanced phosphocrab in those livers. And these effects are dependent on glucagon receptor, as if we perform these experiments in glucagon receptor knockout mice, so lacking glucagon receptor from the liver, provided by Timo Müller and originally from Daniel Drucker, or in mice injected with IUB 396, a glucagon receptor antagonist provided by Brian Finn and Novo Nordisk, we did not observe any of these changes. So we went on and wanted to understand why is depletion of VPS37A affecting gluconeogenesis. And for this, we proposed a model in which usually the glucagon receptor is internalized, from where it still continues to signal from endosomes, but is then ultimately degraded. However, when we deplete VPS37A, the localization of the glucagon receptor is shifted to early endosomes from where the receptor continues to signal, which leading then to enhanced CAMP pKa phosphocrab axis activation. In order to study this, we were a bit halted, as the currently available glucagon receptor antibodies are not so feasible to be used in trafficking studies to actually follow the receptor within the cell. So for that, we teamed up with Oliver Plettenberg from Germany, who provided us with a fluorescently labeled glucagon analog, a Cy5 glucagon, which we of course tested for its functionality, so it is able to activate downstream signaling of the glucagon receptor, which we then use to treat primary hepatocytes on ice, which is blocking all internal trafficking. And this revealed the surface levels then of the glucagon receptor, which turned out to be significantly decreased. Using the same probe in an uptake assay, treating hepatocytes with it and then letting it take up the Cy5 glucagon mediated through glucagon receptor internalization, showed that there's enhanced localization of the glucagon receptor labeled by Cy5 glucagon within the cell. And doing a co-uptake of the Cy5 glucagon with fluorescently labeled EGF to label endosomes, we found enhanced co-localization of the glucagon receptor with endosomal markers, indicating that it is indeed significant enrichment of the glucagon receptor within early endosomes upon depletion of VPS37A. So next, we wanted to understand how the altered localization of glucagon receptor because of depletion of VPS37A is ultimately leading to changes in signaling outcomes. And for that, our collaborators, Aron Novikov and Timo Müller, performed a G-alpha-S recruitment assay in HEX cells, where they found that there's a prolonged recruitment of G-alpha-S subunits to RUB5 positive early endosomal membranes, meaning that there's a prolonged activation and prolonged signaling from the glucagon receptor from endosomal membranes. And this is in line with primary hepatocyte data, where we stimulated those cells with a pulse of glucagon and then followed for crab phosphorylation over time, where we found a reduced phosphocrab deactivation and a sustained signaling of the glucagon receptor from these endosomal membranes. So in conclusion, I've shown you that VPS37A is downregulated in obese and diabetic patient livers, and that knocking it down in the liver is resulting in increased gluconeogenesis, as well as gluconeogenic gene expression and signaling along the CAMP-PKA phosphocrab axis. When we knock down VPS37A, the glucagon receptor is localized to endosomes, from where we have a preferential signaling to glucose but not lipid metabolism. And I haven't shown you this so far, but you're all very much aware, and we've heard this several times before, that glucagon receptor is not only leading to gluconeogenesis, but is also activating beta oxidation through PLC and IP3. However, when we now deplete VPS37A, we have enhanced localization at the endosomes from where it exclusively signals down the CMPPKI phosphocrab axis to gluconeogenesis. Data that I haven't shown due to time constraints from mice fed either chow or high-fat diet revealed that VPS37A had no effect whatsoever on lipolytic gene expression, beta oxidation, CAMK2 activation, or AMPK activation. So in summary, we show that the glucagon receptor signaling can be uncoupled, that the endosomal signaling is, can mediate only glucose production but not beta oxidation. And with this, I'm at the end of my talk. I'd like to thank you for your attention. I'd like to thank everyone involved, especially a former postdoc from our lab, Revati Zekar, whom I'm sharing first authorship with on the recently published paper on this, as well as Anya and all collaborators, some of whom I've already mentioned throughout the talk, and I'm happy to take questions.
So we have a couple of minutes left for questions, and we have a question over here. Michael is coming down with the microphone. It's on the third row.
Thank you for a nice talk. So you had no effect on beta oxidation, and then you still had effect on gluconeogenesis. So compared to the talk we heard just before when we showed the beta oxidation actually fueled the gluconeogenesis, how do your data fit into that perspective?
Yeah, that's a good question. We've seen no alterations in gene expression, like lipolytic gene expression, for example. That's a good question. I don't think I have an immediate answer for that. I'm sorry.
We have time for one or two questions. Jonathan Campbell at the end.
That was a great talk. Do you have an idea of the glucagon receptor interacts with arrestins and how that may be dictating sort of the bias signaling that you're seeing?
Yeah, we've actually were able to rescue all these effects that we have when we do a double knockdown of VPS37A and beta-arrestin, which is then shifting. So if we knock down beta-arrestin, the receptor cannot be internalized, and then it's relocalized just to the plasma membrane. And doing that, we have again effects on lipolytic gene expression and stuff. So if we do a double knockdown of both, the effects that I just presented are gone. Yeah.
Which arrestin isoform, or did you do both?
We knocked beta-arrestin 2. Yeah.